Hi, bioinventories are essential for biodiversity conservation. These inventories can be made at species level or genetic level. In this video, I am going to talk about species level inventories. Species level inventories are not a list of organisms present in an area. It includes more than that. A species level inventory contains taxonomical and ecological information at a species level. The taxonomical information include how the organism is classified, what is the nomenclature, how it is named, and what is the phylogenetic origin of this organism. Ecological information is basically what are the ecological roles played by this organism. So it is not just a list of organisms, it's more than that. By looking at the inventory, we can actually say what are the roles of different organisms played in an ecosystem. So that will be very useful for conservation efforts and to preserve ecosystem function. So the field of science which actually made this species inventory is called biosystematics. You have already know the systematics or biosystematics. Biosystematics is basically the study of biodiversity and its origins. So biosystematics have two different uh, levels of study. Just like I said before, first one is taxonomic in nature, secondly the ecological studies. So biosystematics in taxonomic level, what it do is it study or discover a new organism, and once they find a new organism, they have to identify it to see that whether the organism is a new organism or already identified organism. Then they are supposed to describe the organism in terms of it is a big organism, the anatomical description, how physiologically it is varying, what is its then life cycle. So the full description is required. If it is a microorganism, the biochemical properties, microscopic properties, or the growth properties, everything need to be described. Then naming and classification. To scientifically name an organism, we should know the phylogenetic origin of the organism, how it is classified. The systematic classification is based on the evolutionary origin. So only phylogenetic analysis will help us to name the organism. So we should identify the family, taxa, like class, genus, species, etc. So the systematic classification is required, and should be catalogued so that we can identify this organism easily. So that's the taxonomic part of biosystematics. Secondly, it has an ecological part. In ecological part, we should record the diversity of organism within a species, how much different types of organism or subspecies, subpopulations are seen in this particular species. Life history. Life history is basically the evolutionary origin, how it is originated, what are the close relatives, and how spread this organism is. Everything need to be analyzed. Living habits. Living habits, including the life cycle of the organism, and the habitat. Studies about the habitat also should be done. Role in an ecosystem, that's very difficult because most of the organisms play a wide variety of roles in an ecosystem. So a very detailed study can only find all roles of an organism in an ecosystem. And spatial and geographical distribution, how it is distributed all over the area, that need to be studied. So species inventories are very difficult to make because we should be classify the organisms systematically, then mean we should study the organism in an ecological perspective. We have already identified so many organisms and made inventories for that, but current inventories are very limited in estimating the species richness. There are different reasons are there for it. First thing is the species inventories are very biased. The thing is that when we look at the species inventories or the list of organisms which is already identified by humans, uh, we have identified only best known groups, like the mammals. We have identified almost all mammals in the world. We have identified almost all birds in the world. But when we look at some other species like the bacteria or archaebacteria or insects or some small plants or algae, things like the groups like that has been avoided, largely avoided. So the diversity is highly biased. We are studying only about a small group of maybe large organisms only. We are avoiding vast majority of uh, the small organisms. So our awareness about this species richness is very biased to a small community, and there are best known regions. The biodiversity in some regions are very well studied, like the Northern Europe, which is known as the best studied area in the world. The biodiversity is more or less unknown, known in Northern Europe. But if you look at the other areas like the deep oceans, we don't have much data, or the Arctic and Antarctic, the data is very, very limited. Or even the Amazon, it's a big forest, but the information about the biodiversity is largely unknown. Most of the mammals in the Amazon forest is still need to be described and classified. So, current estimate of species richness is very limited because of the bias. We are studying very limited area with very limited species, maybe very limited type of species. We are avoiding most of the biodiversity, and there is a very high uncertainty with respect to the microorganism, especially if you think about the archaebacteria, which is a biggest group in microorganisms. We don't have enough information about the properties of the archaebacteria, and we know only about very few archaebacteria in the world. But as we know, there is a three domain classification. If you look at the three domain classification, there is prokarya, archaea, and eukarya. The group Archaea is bigger than the all eukaryotes. If you take the algae, fungus, plants, animals, insects, everything together, the number of these all eukaryotes will be less than the number of archaeas present in the world. So, but if you think about the number of archaebacteria identified so far, it is so less. So, the microorganism in any ecosystem which plays a major role, but its identities are largely unknown. Uh, if you look at the overall inventories, the inventories that the temperate region is more complete than the trophic region. Temperate region actually has less number of less biodiversity, but the tropical region is having more diverse biodiversity. So that's the one reason it is inventorying everything in the tropical region is very difficult, because there are huge varieties of species is there are there, but the temperate region has only limited number of species, so inventorying is more easy. That's why the inventories of the temperate region is more complete than the tropics. So, in order to make species inventories, one of the best method is to ATPA, which is all taxa biodiversity inventories. So, as the name implies, it is actually an inventory of all taxas, which means it is including the organisms at all levels, like plants, animals, birds, insects, everything. But mostly, most of this ATBA avoid microorganisms because it is very difficult to study. Maybe they include microorganisms, but the studies will be very limited. The studying about the virus, bacteria, and everything is very difficult. So basically, we study only about the microorganism, which is either too beneficial for us or too harmful for us. So we are avoiding most of the microorganisms which play a critical role in ecosystem. Uh, so ATBA include identification and documentation of all species living in a defined area. So we can define the area, and we are actually studying the every species which are present in that area. So it include identification and documentation. We should identify the organism based on the systematic classification, and also we need to document its ecological properties. So ATB also include the comprehensive tree of life, which shows the relationship between different organisms in a particular region. This particular region can be a forest, can be a country, can be a particular ecosystem, can be a river, or anything like that. So we should identify the organism, we should document the organism, and we should make a comprehensive tree, which shows the evolutionary similarities between these organisms. Further, the ATPA including ecological responses and interactions, ecological responses against different organisms, or the relationship between different organisms. If you think about that, how it is participated in the food web, what is the position of the one organism related to other organism in a food web, and how it is interacting with the biotic and abiotic factors. Those all things to be documented in an ATPA. Then distributional information, both the spatial and temporal distribution of the species should be documented in an ATBI. And this ATBI creation can be either a one point in time inventory, or a year-long study for all seasonal aspects. If you go for a one point in time inventory, we are studying the species richness or the inventory only a particular time in a year. So we may miss some of the seasonal variations among organisms, but in an year-long study, we are considering all seasons. So which is more comprehensive, but it will take more time. Creating ATBI is a bit difficult because very large number of organisms, and most of these organisms are undescribed, as we know. Maybe 90% of organisms is not yet discovered and described. So most of the organisms are undescribed. Even if we know about some of these organisms, they are even identified, but taxonomically we haven't classified the organism. So it's a bit difficult to to include it in the inventory as a separate entity or a subspecies of another entity or something like that. Uh, Next, making the ATBA, the genetic information is needed. Acquiring genetic information is difficult because it's expensive, time consuming, and tedious. So, the genetic difference among the organism is a bit of a task. And next, the in an ATBA, we should have the ecological aspects of the organism. Most of the organisms has a complex ecological roles and relationship. Most of these relationships are very difficult to study. So making an ATBA is basically very difficult. So, and these organisms has multitudes of relationships. It has interacting with the biotic and abiotic factors in different ways. So there will be different types of interaction among these organisms and abiotic factors. So including everything in a inventory is actually very difficult. So some of the countries has initiated national all taxa inventories, which is to document all the available bioresources in an inventory. To document, identify and document every bioresources in that country uh, in that inventory. And the work for that is actually progressing in most of the countries, but it is largely partial. And most complete inventories are of the Swedish taxonomy inventories, which is known as the most complete or comprehensive inventories so far, but it is not. If you think about the completeness, it is most complete one, but it is absolutely it is not. Maybe 50 or percent of the biodiversity is still missing in that inventory. If you think about this situation, like in India, it's a huge country with huge biodiversity and resources. It's very difficult to make a comprehensive national all taxa inventory. So it's generally observed that the full inventory is nearly impossible in near future because of the huge number of organisms, and most of these organisms are undescribed. So people are always trying for it, but right now for inventory purposes, what we do, we have two different strategies. First one is taxon specific. Means that we are not going to study, when we are making a species inventory, we are not going to study all type of organisms. We are studying only a small group of organisms, and we are making conclusions based on small group. That is called a taxon specific inventories. For that, we basically select the large organisms, which can be easily find in a area. If in a forest, if you it is easy to find a elephant in a forest, but if you are looking for an insect, it will be very difficult. So the taxon specific inventories for large organisms, or maybe if you want to find a tiger, it will be much easier than you are going to finding an insect or a microorganism. So in one strategy is to make inventories only for the large animals, large organisms, plants and animals. So it will be much easier than making inventory for other organisms. And the second strategy, which is more accepted strategy and a more scientific way, is to make inventories in such a way that we are studying some of the areas in detail, and based on this studying this limited area, we are projecting or estimating the biodiversity in other areas. So basically we divide the whole area into two. In some areas we will conduct a detailed study. In other areas, we will do only a peripheral study, and we make to estimate the biodiversity in those areas. So that's the second strategy, and I am going to explain the second strategy more detail. So this strategy has two different sites: intensive sites and extensive site. In intensive sites, we will conduct intensive studies. We will study the complete biodiversity in that area, including all species, including the microorganism, including the insect, including the animals, plants, and everything. This should be a detailed study. By this study, what we do, we identify the key organisms or the indicator organism. So by studying the biodiversity in detail, we will get an idea which are the indicator organism. If we find the indicator organism, this we can actually get an idea about all biodiversity. Suppose if our indicator organism is a super predator like a tiger, in order to support a tiger in a ecosystem, there should be enough herbivorous organisms there. So in order to support that much herbivorous organism, enough plants will be there, enough productivity should be there. So if you just look at the diversity or the species richness of tiger population or the or related populations, we can actually get an idea about the overall biodiversity. So in this strategy, in inventorying, we have intensive sites and extensive site. In intensive sites, we will conduct a detailed study. In this study, we will analyze the biodiversity at all levels, and by this study, we will identify the indicator organism. Okay, and we will make estimate the relationship between the number of indicator organisms and overall biodiversity. In extensive site, we study only the presence and number of the indicator organism, and based on that number, we will project the number of overall biodiversity of that region. So that's a process. So the overall region, maybe if you are going to study a large forest like Amazon, you cannot go and study every parts of it. So what we do, we will separate the regions into extensive sites and intensive site. Intensive sites will be very few in number, maybe 5-10 sites, and all other area, most of the area will be extensive site. And in intensive site, we will study the detailed analysis, and based on that data, we will project it into the all extensive site. And study in this intensive site and extensive site is very different. So in an intensive site, we'll go for intensive data collection techniques. We want to capture everything, every possible biodiversity data. And the sampling methods should be taxonomically inclusive for everyone. So actually the identification of the large plants and animals is easy, but if you want to identify all the insects, that will be a bit of a challenge. Or identify all type of microorganism, that's also a challenging thing. So the sampling techniques will be inclusive, and the data will be collected both quantitatively and qualitatively. Both type of data will be collected in an intensive site, and we will make a complete species inventory for that intensive site. Okay, and based on that complete species inventory at intensive site, we will actually calibrate the quantitative data. We are actually finding some of the indicator organisms which directly indicate the biodiversity in the area. The indicator means if the number of that indicator organism increases, there is a parallel or parallel increase in the overall biodiversity. If the number of that particular indicator organism is reducing, there is a proportional decrease in the overall biodiversity. That's what an indicator organism should do. So we actually calibrate the quantitative data by quantitative sampling, and estimation protocols will be established, and we will make some ways to extrapolate these results to other regions like other extensive sites. And then sometimes we will do supplementary sampling at intensive site to calibrate again, recalibrate the data. Inventorying at extensive site is different. In extensive site, we only identify look at the indicator species. In intensive site, we have looked at the all biodiversity, but in extensive site, we are looking only at the indicator species. and we capture information only about these focal groups. The indicator species may not be a single species. Sometimes we will find an indicator at different trophic levels, so we will get a better data. So that's what called a focal group. Maybe a group of organism, maybe two or three organism, or five or six organism will be made as a focal group, which together act as an indicator of the biodiversity. In this site, we are not going for intensive sampling. Instead of that, we are using quantitative sampling. We actually want to know the quantity, how much number of this indicator organism presents, and standardized quantitative methods will be used. And species richness and diversity is actually estimated based on the indicator organism. We are not directly estimating the species richness and diversity. Instead, we are making an estimate based on the number of indicator organisms. So, thank you so much.